Okay, I think it's now getting to time to kick off this webinar. So I want to first off thank everyone for attending. And I want to also point out that if you have any questions during the seminar, you can please type those questions into the Q&A box or into the chat function. If I can, I'll answer them during the presentation. And if not, I will moderate a Q&A at the end of this where I'll read the questions and then Todd and I will answer them. So I'd like to start. I'll introduce myself. I'm Travis Block. I'm the Chief Technology Officer at StemBioSys. I've been with the company for 6.5 years. Prior to that, I was working with the company's core technology in our scientific founders lab at the University of Texas Health Science Center at San Antonio. And the work that's going to be presented today by my colleague, Dr. Todd Herron, is work that we've collaborated on going back to 2018 or 2019. So we've been working together on this work for 4 or 5 years, and it's resulted in a number of publications as well as the commercialization of several technologies that I think are going to be really interesting to everyone on this call. And so with that, I'm going to begin by giving a brief overview of our CELLvo Matrix technology before handing it off to Dr. Todd Herron, who's the director of the Frankel Cardiovascular Regeneration Core at the University of Michigan, and a real thought leader and innovator in the space of cardiotoxicity testing in vitro. So again, thank you all for joining our webinar this morning titled High Throughput Cardiotoxicity Screening Using Mature Human-Induced Pluripotent Stem Cell-Derived Cardiomyocytes. So I always like to begin these presentations by giving just a little bit of history. And so these faces that you're looking at here, Alexis Carrel and Montrose Burroughs, are actually the inventors of modern cell culture. And they didn't set out to do it on purpose. Alexis Carrel was a transplant surgeon who actually won a Nobel Prize for developing techniques for vascular sutures that enabled organ transplants. But in his lab, they were taking muscle tissue sections and putting them onto microscope slides to study capillary bed structure. And when they were doing that, they saw that these cells would migrate out of the tissue and stay alive on the microscope slides. And so they found that they could continue to bathe these cells in a nutrient bath and maintain their viability. And so they published this method all the way back in 1911.
They published a paper where they described.
Sorry, I don't know how that audio got in there. But in 1911, they published the methods that were the first ever methods for the culture of animal cells. Obviously, bacterial cell culture had existed for centuries with those sourdough cultures that everyone was doing during the pandemic lockdowns and with people making beer and wine. But this was the first time that anyone had ever been able to maintain the viability of animal cells outside the body. And so what's the problem with this? The problem with this is that they didn't set out to do this on purpose. And so they ignored key aspects of how these cells exist in the body. And so what you're looking at here is an SEM image of a cell in its native microenvironment. And when you take a cross-section of this image, you can actually see, going from the top left to the bottom right, a phospholipid bilayer. And so what you're seeing is actually the boundary between a cell and its extracellular matrix. And until it's labeled, it's very difficult to tell where one ends and the other begins. And that's because in reality, cells are interacting with ECM components directly across their the phospholipid bilayer. ECM components are plugging in directly with cytoskeletal components. And what you're watching here, you're seeing this image, dynamic reciprocity between the cell and its ECM in action. The cells are elaborating their matrix, and that matrix is in dictating cell function by interacting directly across the cell membrane. So there is just too much communication going on across this membrane to discount it in culture. Yet, that is what we do with traditional culture methods. And over the last 2 decades, really, the extracellular matrix and its role in dictating cell function has gotten a lot more attention, and there have been a lot more publications looking at how the microenvironment regulates cell function. However, most of these studies have been extremely reductionist. Kind of the classic way of looking at this is the study in the top left corner. We started having studies where people were taking acrylamide gels. They were varying the ratios of acrylamide to bisacrylamide to adjust the stiffness, adding in some RGD peptide so the cells could stick to it. And then they were studying what stiffness is best for cell proliferation, what stiffness is best for self-renewal, what stiffness drives osteogenic differentiation. And then you'd get papers that say, oh, the correct stiffness for osteogenic differentiation is 100 kilopascals, or whatever they would say. But this isn't really true because these are still extremely artificial environments. And so what you're really getting the answer to is what stiffness is best when you are on a totally flat substrate with a single binding protein. And so then some studies started to try to get at these differences. And the perfect example of what shows how overly reductionist these are is this paper that we have right down here at the bottom, published in PNAS in 2010. And so what they did here is they made little cookie cutter cutouts in the bottom of tissue culture plastic dishes. And so the stiffness was exactly the same. The biochemistry of the substrate was exactly the same. And the only thing that they changed was the architecture, such that it forced cells to take on a different morphology. And when they forced cells to take on a different morphology, they responded differently to the substrate, and it changed gene expression pathways and could drive differentiation down adipogenic or osteogenic differentiation pathways. So this is incredibly important because but not for the finding that cell shape dictates differentiation, but more so for the finding that there is a dynamic interplay between all the aspects of the extracellular matrix. So the cell-cell interactions, the cytokines, so the soluble factors, the structure, the mechanical factors, the biochemistry, how proteins are being presented within a matrix. Is the matrix also storing growth factors for the cells? All of these things are critical to determining cell fate, and none of them can be studied in isolation because they influence one another. And so at StemBioSys, we take what I think of as a more humble approach, where we try to be a bit more biomimetic. And so what we do is we take matrix-producing cells, we put them into a dish, we allow them to grow to confluence, and then we induce them to secrete a matrix just by adding extra vitamin C to their culture medium. And then we remove the cells once they've secreted a matrix using a non-ionizing detergent. And we wash that thoroughly, and then we air dry it onto substrates so that all our end user has to do is add PBS to rehydrate. And so what we're doing here is we're producing a protein, glycoprotein, triglyceride-rich matrix that has distinct architectural and physical and chemical properties based on the cells used to produce it. And these matrices elicit more biologically relevant phenotypes relative to traditional cell culture. And this method is highly reproducible, highly scalable, and we're able to do it in a wide variety of formats. So what you're seeing here on the left-hand side is an SEM image of the matrix. In the center panel, you're able to see that actually just in bright field, you can make out the structure of the matrix. So this is a matrix that's highly fibrillar. And so you can see the fibers that are oriented in a single direction in this image. And then in the right-hand panel, you see that you're still able to do bright field imaging of cells in the matrix. You can still do fluorescent imaging of cells in the matrix. You can do confocal, you can do high content imaging. And when we ship these matrices to our customers, it looks like you're just receiving tissue culture plastic. When you hold it up to the light just right, you can see a thin film that it almost looks like if soap dried on a plate. And what that is that's just the air-dried natural hydrogel that you can add PBS to rehydrate and then seed your cells. And we're able to make this in everything from hyperflasks down to 384 well plates. So we can do it in high throughput formats as well as cells for bulk producing, or as well as vessels for bulk producing cells. But what's really kind of the salient feature of these matrices is not the ease of use, though it is extremely easy to use. The real salient feature is that it induces more biologically relevant phenotypes. And so what you're looking at here in this bottom right panel is a gene expression array where we show that cells that are cultured on our matrices have gene expression that is more similar to cells that are freshly isolated versus cells that have been cultured on this, I believe, was fibronectin here. And that what the appropriate control is going to vary for different cell types, but we've been able to validate this approach works for a wide variety of tissues. But now when I'm talking about our ability to better mimic microenvironments to elicit biologically relevant functions, a logical question to ask is, well, what cell types does this work for? Because there's a lot of different microenvironments in the body. Traditionally, cell culture microenvironments have generally been one-size-fits-all solutions. People will coat fibronectin for all sorts of cell types. They'll throw down laminin for all sorts of cell types. They'll use Matrigel for all sorts of cell types, or just tissue culture plastic. And the reason that we've gotten away with that is that we've traditionally had extremely low bars for judging efficacy of cell culture substrates. Traditionally, people say that a substrate works if cells stick to it and remain viable. But I like to say that this is a really painfully low bar because there's a whole lot of room for variability in cell function between normal healthy function, that is what they would do in the body, and death. And so what we're trying to do is elicit that actual normal healthy function they have in the body and not be content with cells that are attached to a surface that just aren't dead. And so what we found is that we can actually take structural cells from different tissues in the body and put them into the culture environment and go through our process of making ECM, and that those extracellular matrices retain properties of the tissue of origin of the cells used to produce the matrix. In doing so, we're able to produce a variety of tissue-specific microenvironments. Now, I need to be careful to mention here that while we talk a lot about tissue specificity at StemBioSys, there's also common motifs in tissues. And so because of that, we don't expect that we'll have to make 100 different matrices for 100 different cell types, but we're continuing to expand our tissue-specific offerings to better approximate more microenvironments in the body, so that we can elicit biologically relevant phenotypes from a wide array of cell types from various tissues. And again, I mentioned that we validated this approach on multiple systems. The first paper that we published on the tissue specificity of these matrices was back in 2016. When we looked at matrices produced by different types of mesenchymal stem cells, we showed that cells derived from isolated from bone marrow made a matrix that was 100-fold stiffer than cells isolated from adipose tissue. And in turn, even though the biochemical composition was extremely similar, the bone marrow derived ECM supported an induced osteogenesis, whereas the adipose derived matrix induced adipogenesis. Similarly, in a 2019 paper, we showed that we could produce a matrix from articular chondrocytes that better maintained a chondrogenic phenotype from articular chondrocytes, so that when we put them into a pellet culture, they would make a collagen II and GAG-rich matrix, versus cells isolated and cultured with traditional methods that basically lost all of their attributes of chondrocytes in the body. They're no longer making GAGs, they're no longer making hyaluronic acid, they're no longer making collagen II, they're just making scar tissue in a dish. And so those have been really interesting findings. We actually have an active NIH SBIR grant right now looking at this application, where we're using a perinatal stem cell derived matrix, actually the same matrix that we use for maturing cardiomyocytes, for the isolation and culture of human islets. And we've shown that it dramatically extends the culture time that you can maintain human islets with healthy function, and that it improves their glucose responsiveness such that they're in a glucose stimulated insulin secretion assay, they respond much more strongly to glucose, but still come back to a nice baseline when it's removed. We've just recently launched our neuromatrix, which is an astrocyte-derived matrix that supports a variety of different neuronal cell types, as well as neural support cells like Schwann cells, glia, astrocytes. And then what Todd is going to go into a lot of detail on today is the application of CELLvo Matrix Plus for the maturation of human iPSC-derived cardiomyocytes with specific applications in cardiotoxicity testing. So before I hand it over to Todd, I'm just going to briefly give the key takeaways on our technology. And so at StemBioSys, we have a suite of tissue-specific matrices and primary cell products that are currently being sold as research tools. These tissue-specific extracellular matrices have properties that are typical of the tissue of origin of cells used to produce the matrices. And so, again, that's not just the biochemistry, but it's the architecture and the mechanics of it, as well as the biochemistry. So we try to make these as biomimetic as we can by not disturbing the matrices that these cells produce in vitro. And these matrices, in turn, drive biologically relevant phenotypes of primary and stem cell-derived somatic cells cultured on the matrices. And CELLvo matrices induce rapid and spontaneous maturation of iPSC-derived cardiomyocytes. That's the data that Todd's going to be getting into. And I'll bring this up again at the very end, but if you have scientific questions, you can feel free to email me directly at travis.block@StemBioSys.com. And if you're looking for product quotes, you can reach for our director of sales, Iris Flores, at iris.flores@StemBioSys.com. And with that, I'll hand it over to my friend and collaborator, Dr. Todd Herron of the University of Michigan, to discuss some of his most recent results using CELLvo Matrix Plus for the maturation of iPSC-derived cardiomyocytes. And Todd, you're on mute.
All right. Hello. Thank you, Travis. Thanks for the introduction. I hope you can see my slides now. You can see the title page. So today I'll be discussing data that my laboratory has developed over the last 5 or 6 years, working with CELLvo Matrix Plus, which is a matrix that Travis introduced, that we use to mature the phenotype of cardiomyocytes. So today I'm going to be presenting data on high throughput cardiotoxicity screening using mature hiPSC-derived cardiomyocytes. And the talk is broken up into essentially 2 different parts. One part will go over the maturation of the cardiomyocytes and the use of these mature cardiomyocytes for acute drug response data analysis, and that is exposure of a drug from 30 minutes to 1 hour. And then I'll also present data that we've collected more recently, developing methods for measuring the chronic responses of the cardiomyocytes to drug exposures. And for that, I'll be introducing work that we've done utilizing a genetically encoded calcium indicator called GCAMP6. So for introduction, so now in 2023, the human cardiomyocytes have been validated by an enormous international validation effort led by the Health and Environmental Sciences Institute and the FDA, and the cells have been validated for use in detecting drug responses and adverse electrophysiology events to drugs. The validation has been so successful that now human cardiomyocyte data is being employed for FDA regulatory decision-making. And this was published recently in 2022 in the journal Toxicological Sciences, in an article called The Use of Human hiPSC-CMs in Non-Clinical Regulatory Studies for Cardiac Safety Assessment. The assays that have been validated include multi-electrode array assays, the use of optical dyes for sensing the voltage changes and the membrane changes and the action potential, and also using calcium sensitive dyes. Currently, the cardiomyocytes are being validated for detection of chronic effects of compounds and medications, with exposures lasting from 1 hour and extending out to 4 days of chronic exposure. And this is in a newer blinded validation study, again led by HESI. The assays being used for electrophysiology again include the MEA assay, which is attractive because it offers repeated measures However, the MEA assay offers very low spatial resolution of data, where there are only 8 recording electrodes per well of most 96 well plates. And it's known that in these wells, not all of the electrodes will produce data, and oftentimes a single electrode is selected. So we have a hypothesis that repeated measures using optical mapping for chronic assays offers a higher resolution assay, but requires validation. So the movie at the bottom right illustrates the difference between the data collected using MEAs, where data would be collected where the electrode terminals are located, here 4 different locations. Using optical dyes, you can see that there's a lot of information and a lot of data that's missed and not recorded when using MEA approaches. So limitations that our research has addressed in recent years, one is cellular maturation, and it's been pointed out broadly in the field that there's a need to advance the phenotype to more adult-like phenotypes. 2 is developing a high throughput optical mapping device to enable repeated measures using genetically encoded calcium indicators. And finally, the issue of chamber specification and the development and use of atrial cardiomyocytes versus ventricular specific cardiomyocytes. So in 2020, we published a report on maturing cell cardiomyocytes, shown here, going from cryopreserved cells, cryopreserved vials of cells. We can now plate a density of 75,000 cardiomyocytes per well. Of a 6-well plate, these plates are obtained from StemBioSys, and they come pre-coated with Matrix Plus ECM coating, which promotes the maturation of the cardiomyocytes. The maturation effect is pretty rapid. Following plating the cardiomyocytes on Matrix Plus, after 7 days, we routinely observe the appearance of rod-shaped cardiomyocytes, and upon staining for sarcomere proteins, here for troponin T and in red for alpha-actinin, we see much better sarcomere structure and myofilament organization when cells are maintained and cultured on Matrix Plus ECM. At the bottom, you see 3 different images. On the far left is troponin I staining and confocal imaging over a large field of view, just to show that the rod-shaped morphology of the cells is throughout the monolayer. In the center, these again are cell cardiomyocytes maintained on Matrix Plus ECM, and you can see the robust maturation inducing effect, where nearly 100% of the cardiomyocytes are now rod-shaped rather than circular in shape. And sarcomere analysis shows significant maturation of the cardiac myofilaments. We also have done some characterization of the maturation of the mitochondrial function to look at metabolic maturation of the cardiomyocytes induced by the Matrix Plus human ECM. Here, this is a comparison of cell cardiomyocytes plated on Matrigel, which is a commonly used mouse-derived ECM. And you can see the cells plated on Matrigel maintain a circular morphology, a circular shape throughout the plate of cells. Whereas on the other hand, the same batch of cardiomyocytes plated on Matrix Plus, the human ECM product from StemBioSys, the cardiomyocytes all become rod-shaped. And when we do analysis, here the staining is for the mitochondria using JC1 mitochondrial specific dye. And this dye, the way that it works is in highly functioning mitochondria, the dye will shift from green fluorescence to a red fluorescence, and the ratio of the red to green fluorescent signal is an indication of the membrane potential of the mitochondrial membrane space. And the more hyperpolarized the mitochondria are, the healthier they are expected to be. So here we can see that the Matrix Plus ECM induces a significant maturation of the mitochondrial function, showing increased polarization of the inner membrane space of the mitochondria compared to the same batch of cells plated on Matrigel. This is a closer examination of the maturing effect of Matrix Plus on the cardiac sarcomere. Here, this is staining for alpha-actinin in red with high-resolution confocal imaging in the 2D monolayer. And you can see the cells maintained on Matrigel, the mouse ECM. Although they do have sarcomeres, those sarcomeres are very loosely compacted and very fetal in their appearance and their phenotype. On the other hand, the sarcomere structure of the cardiomyocytes plated on Matrix Plus, we see rod-shaped morphology of the cells, and we also see very good organization of the cardiac sarcomeres throughout the length of the cardiomyocytes, where there's one axis of sarcomere formation, which is much more reminiscent of the mature adult cardiomyocytes. And when we analyze the average sarcomere length for the cardiomyocytes cultured on Matrix Plus, they have a much longer resting sarcomere length relative to the same batch of cells plated on Matrigel. We also have done some work using other iPSC lines and other cardiomyocyte sources besides the Cellular Dynamics Fujifilm products. Here, this shows the quantification of the circularity index calculated from phase contrast images and showing paired analysis for each cell type. So each cell type plated on Matrigel is represented with black symbols, and the corresponding or same batch of cells plated on Matrix Plus is shown in red symbols. And for for 4 different iPSC lines and cardiomyocyte sources, we can see robust induction and reduction of the circularity index of cardiomyocytes from multiple sources and multiple cell lines, indicating that the maturation inducing effects of the Matrix Plus ECM can be applied broadly to probably any iPS cardiomyocyte cell line. So once we were able to robustly mature the cardiomyocytes, next we built a high throughput electrophysiology plate reader system. We were fortunate to receive NIH SBIR funding from the National Institute of Environmental Health Sciences that was given to our startup company called Cartox, which was purchased by StemBioSys, and now the commercialization of this plate reader is being done by Curie Bio under the name Nautilus. So the plate reader, it can accommodate multi-well plates shown here, fed into the plate reader. And this enables whole plate electrophysiology analysis. The movie that you see playing at the bottom left, these are spontaneous calcium transients traveling through each individual iPS cardiomyocyte monolayer. And the data is collected and analyzed in parallel, which enables very high throughput, rapid and robust data collection. And using calcium transient dyes as a surrogate marker for contractility, it enables examination of the calcium transient amplitude as an index of contractility to detect differences of contractility. We've done validation of the Cartox high throughput EP plate reader, which is capable of measuring voltage changes or action potentials using a dye called FlowVolt. And also separately, not simultaneously, we can also record calcium transients using a dye called Calbright 520 AM. So panels A and B, these are representative traces of atrial cardiomyocytes spontaneous beating activity. A shows spontaneous action potentials, and panel B shows spontaneous calcium transients recorded by our high throughput plate reader. We compared the data collected for action potentials, denoted by a VM with purple symbols, and compared that to data from the same plate where we recorded calcium transients, shown here in the blue-green symbols. So the beat rate we found to be similar when we measure either the action potentials or the calcium transients. The conduction velocity is very rapid compared to immature cells, here around 40 to 50 centimeters per second, which was similar whether we recorded the action potential traces or calcium transient fluxes. In panel E, we quantified the action potential duration and the calcium transient duration, and it's well known that the action potential duration should be much shorter than the calcium transient duration, which we observe here. And finally, we also quantify what's called action potential triangulation and calcium transient triangulation, which is a reflection of the duration of each of those events. And similar to the way that the duration is longer for the calcium transient, also the calcium transient triangulation is, as expected, greater than the membrane potential triangulation. So that data, we can also represent it using heat maps. Here in panel G, on the left is a heat map indicating the action potential duration 30. And on the right indicates the calcium transient duration 30 recorded from this plate of cells. And you can see the difference in colors indicate differences in those durations noted by the numerical quantification on the right. Similarly, in panel H, a duration 90 heat map also shows that the action potential duration is much shorter in duration than the calcium transients. And finally, because the triangulation index is a combination of the duration 30 and duration 90, you can see that the action potential triangulation is much smaller than the calcium transient triangulation. Finally, we have validated the cardiac EP plate reader for acute responses to compounds with known cardioactive effects. In the top panel here, this is the response of atrial myocytes to isoproterenol at a dose of 100 nanomolar. Panels B through E quantify the before and after isoproterenol. So in panel B, we see the isoproterenol increases the beat rate. In panel C, increases the conduction velocity of the monolayers. In panel D, increases the contractility as a reflection of the increase in calcium transients. And in panel E, there's a modest effect on the duration of the calcium transients. In the next row, we validated the cells, the mature cardiomyocytes in the cardiac EP plate reader using a hERG channel specific blocker, here E4031 at a dose of 500 nanomolar. And again, we did a similar quantification in panels G through J, showing that E4031 had modest effect on the beat rates, but had very significant effect to reduce conduction velocity. In panel H and panel I, E4031 reduced the contractility significantly, reduced the calcium transient amplitude, and in panel J, the well-known effect of IKR blockers like hERG, channel blockers like E4031 to prolong the calcium transient duration. And finally, in panel K, we also tested the response to ibutylide, which is an antiarrhythmic drug that has risk to cause arrhythmias. Here in these atrial cardiomyocytes, there was a kind of a neutral effect, maybe increased beat rate induced by ibutylide. And panel M shows a dramatic reduction of conduction velocity On panel N suggests some calcium channel block by butylide with dramatic reduction of the calcium transient amplitude and a reduction, which suggests a reduction of contractility. And in panel O, we see the increased duration of the calcium transient induced by ibutylide, as you would expect. So the next bit of data that I'm going to show you is surrounding the validation of the plate reader for chronic assays. And for this, we turned from using calcium sensitive probes like Calbright to using a genetically encoded calcium indicator referred to as GECI. And we tested the GCAMP6 genetically encoded calcium indicator, which is a fusion protein, fusion of GFP together with calmodulin, linked together by this M13 peptide. The GCAMPs that we tested 3 different variants of GCAMP and delivered them by adenovirus. We tested 3 different variants that vary based on their kinetics to bind calcium. So there's a fast, medium, and a slow GCAMP. We tested their effects on the cardiomyocyte, electrophysiology and function, and suitability for chronic assays. So again, the way that we have done this testing, on day 0, we've utilized cell cardiomyocytes that are cryopreserved and plated them on Matrix Plus coated 96-well plates. We wait for 4 days to allow the cells to mature a bit. And then on day 4, we introduce a recombinant adenovirus that is carrying the GCAMP6 isoforms. Then we wait for a few days while the virus protein, the GCAMP6 protein, expression increases and reaches a steady state. And then on day 7, following delivery, or day 7 of the protocol, then we start doing chronic drug exposures and chronic analysis of the effect of different compounds on the function of the cardiomyocytes. And as I pointed out before, the data that I'll show you, we did a careful validation in order to determine which GCAMP sensor is optimal for repeated measures. We used GCAMP6 fast, medium, or slow. So first we did some microscopy imaging, and by day 2, after delivering the GCAMP virus, we can detect expression and we can detect low-level calcium transients that increase over time. So going from day 2 to day 5 of this monolayer, you can see much more robust expression of GCAMP, which enables recording of much larger calcium transients. And you can see that each GCAMP isoform that we tested, fast, medium, or slow, the fast traces are shown in red, the medium kinetic binding shown in blue, and the slow kinetic binding are shown in green. So all 3 viruses work well, express well, and enable electrophysiological recordings over a period of time, here going from day 2 to day 7. So when we do quantification of the GCAMP signal over time, here plotting the calcium transient amplitude going from day 1 to day 8 after viral transduction and delivery of the GCAMP, we can see stable calcium transient amplitudes beginning at about day 3 and extending to day 8, and correspondingly fairly stable spontaneous beat rates over time. So this suggests that for chronic assays, we should begin the recordings at day 3 after viral transduction, and we can extend at least till day 8, and perhaps even longer if we extend the time of the experiment. So using the high throughput plates and the high throughput plate reader, here we can see quantification of the calcium transients over time. So the GCAMP6F data is in columns A, B, and C. And panel A is day 3, panel B is day 4, and panel C is day 7. So you can see for GCAMP6F, pretty stable looking calcium transients over time. For the medium kinetic binding GCAMP, also we can record from day 3 to 7, and we can see that there's a little bit longer duration of the calcium transient from days 3 and day 4. And with the slow isoform in green, we can see that the calcium transient duration actually is even more prolonged than the 6F and the 6M. And when we quantify the calcium transient duration over the time course from day 3 to day 7 after gene transfer, we can see that indeed at day 3, we can stratify the data by the kinetics of the GCAMP6 calcium binding. So the GCAMP6F calcium binding indicator, GCAMP indicator has a much shorter calcium transient duration than the other two slower binding GCAMP6 isoforms, which changes over time but is maintained up to day 7. Next, we look to see the response of the cardiomyocytes transduced with GCAMP to see their isoproterenol response. And so for GCAMP6F, we observed an expected isoproterenol response where the ISO actually caused a bigger calcium transient, increased beat rates, and shorter duration of the calcium transient. On the other hand, the GCAMP6M isoform caused an abnormal isoproterenol response where we could observe increased beat rates, but we actually saw a reduction of the calcium transient amplitude. Also, when using GCAMP6S, we observed an abnormal response to isoproterenol, where the isoproterenol had a positive chronotropic response, but there was no corresponding positive inotropic response. And that data is quantified here for each isoform, showing positive chronotropy for each of the isoforms. But for the inotropy, or the effect on the calcium transient amplitude, the GCAMP6F caused positive inotropy, and the other two isoforms did not. And finally, with the calcium transient duration, we could see the expected effect on the lusotropic nature, or the relaxation of the cardiomyocytes. So GCAMP6F showed us a response to isoproterenol of positive chronotropy, positive inotropy, and also positive lusotropy. So these are all the expected physiological responses of the heart to isoproterenol. So we were very pleased to see that the GCAMP6F enables a normal response to isoproterenol in our assays. We also did some validation of the GCAMP6 indicator just to make sure that we could also detect hERG channel block when we use the GCAMP6. And indeed, for each isoform, we could see prolongation of the calcium transient duration, also slowing of the beat rates induced by E4031. We could observe a reduction of the calcium transient amplitude only in the case of using the GCAMP6F, and for all three isoforms, we could detect the prolongation of the calcium transient duration that's expected with hERG channel block. So that gave us the confidence to utilize GCAMP6 for chronic studies. And here I'm going to show you some like a case report from our recent participation in the HESI-led blinded validation study. We received compounds from HESI. One of those compounds was BMS 986094, which is a potent hepatitis virus C replication inhibitor, but also unfortunately caused heart failure in patients and was discontinued from use in 2012. We also have tested doxorubicin, which is also another compound, chemotherapy in this case, that can cause heart failure at high doses. So here we employed the same strategy, utilizing cryopreserved cardiomyocytes matured on Matrix Plus. Then we introduced the GCAMP6F virus. And then starting on day 7 through day 10, we did optical mapping studies to determine if we could detect the chronic effects of these drugs to cause cardiotoxicity. The dosing that we used was recommended by the HESC committee and is based on clinical data. We did daily recordings and also collected daily phase contrast images of the monolayers to look at the structural effects of these compounds. So here, this is data from our testing of the cardiotoxic compound BMS 986094. The pictures that you see, the very top column going across is dose 1 with escalating doses going down to dose 3 into dose 4. And each vertical column represents a different point in time. The far left column is time 0 for each dose. The middle column is the image at the 24-hour time point. And then finally, the far right column shows you the same well at the 72-hour time point. And if you focus your eyes here to the final time point at the highest dose, at the 72-hour time point, you can see significant cell death indicated by the cells not being there any longer and lifting off the plate. And that data is quantified in the top right panel. And you can see that every dose starts with a relative confluence around 1, but we can see by day 3 and day 4 of the recording, significant induction of cardiotoxicity induced by the BMS compound, which is known to cause cardiotoxicity. These images are taken using a time-lapse imager in the microscope called the IncuSite zoom. Next, we did this is the data from the functional recordings over time. So each of these parameters are normalized to the time 0 values that were recorded. And so our reflection and our values relative to the start of the experiment. And if we focus on panel B, this is the quantification of the contraction amplitude, which is measured from the surrogate marker of the calcium transient amplitude. And here, for this drug known to cause toxicity, we can see that by 2 days after the treatment, we start to see a reduction in the contractile function, which precipitates over time and gets worse up to day 4, where you can see nice stratification of the doses. So here's dose 1 is similar to the vehicle control, dose 2 is in red, and then doses 3 and 4 here have bottomed out, showing that we can detect the toxicity induced by that compound. Here's another case of our testing for doxorubicin to determine if we can detect the chronic effects of doxorubicin. Here the data is laid out the same way as in the previous example. And again, if you focus your eyes from time 0, dose 1, down here to the extreme case of the highest dose at the final time point, you can see that the cells have pretty much all started to round up and have started to lift off the plates, indicating significant cardiotoxicity. And that's quantified here, where we can see the highest dose of doxorubicin, even by after 1 day of exposure, starts to cause toxicity. And here again, this is the summary of the functional data, looking at the detection of functional cardiotoxicity induced here by doxorubicin. And again, here, if you focus on panel B, which is the quantification of the contraction amplitude, we can see that over time, the contraction amplitude decreases and decreases to a very significant level in the doxorubicin treated groups, especially at the highest doses of doxorubicin, where there's practically no contraction that's still detectable at that time point for that high dose. So finally, I just want to comment now on our work that we've done making cardiac chamber specific cell types. and the impact that that may have on cardiac safety screening and development of new compounds. So in our lab, we utilize a published protocol from JCI Insight in 2018 to differentiate iPSCs either to ventricular cells using small molecules. We can produce high number of ventricular cells and doing a similar protocol, but with the introduction of retinoic acid at days 3 to 6. This induces the cardiomyocytes to take on a more atrial cardiomyocyte specific phenotype. The differences between the two differentiation protocols are highlighted in these two movies. The ventricular cells that we produce consistently contract more slowly and with a bigger contraction amplitude. On the other hand, the atrial specific cells always beat with a much higher spontaneous beat rate, and the cardiomyocytes tend to be smaller. Here's phenotype analysis to confirm the identity of the atrial and the ventricular specific cardiomyocytes. So here we probe for beta myosin heavy chain, which in the human heart is a ventricular specific marker. We can see that there's barely any detectable trace of this ventricular marker in the atrial cells, but the marker is present in high abundance in the ventricular cells. Here in lanes 5 to 8. And in this experiment, since we can separate the cells and then recombine them, we did some control experiments mixing the two cell types together, and that causes much different expression patterns of beta myosin shown in panel B. And when we look at the function of the cells, the atrial cells have a much shorter action potential than the ventricular cells, and the control, the intermediate, where there's 50-50 mixture of the cells, we get an intermediate phenotype, which is also quantified in panels D and E for the duration of the action potential. And this is likely due to chamber-specific ionic current expression. So the question is, what implication would this have for cardiac safety screening and doing drug toxicity? Is that there are chamber-specific responses to some medications. Here, this data shows our testing of a drug called vernaculant, which is used in Europe to treat atrial arrhythmias because it's reported to have chamber-specific effects. Here we can see chamber-specific iPS cardiomyocyte effects to this drug. The data in brown is the atrial specific cardiomyocytes response to vernaculant. And on the other hand, in blue, these are the ventricular cells response to vernaculant. You can see much different responses based on whether we use atrial specific or ventricular specific cardiomyocytes. So for summary and conclusion, one, we're very excited that we can mature the structure and the function of the cardiomyocytes using Matrix Plus in robust 96-well plate formats. The Nautilus or Cartox EP plate reader enables rapid and robust data collection for in vitro toxicology. We've determined that the genetically encoded calcium indicator GCAM6F is the optimal variant for repeated measures for chronic assays. GCAM6F expression, we're able to still detect cardiotoxic effects of medications. And finally, chamber-specific cardiomyocytes can be produced in high number. And the data that we've collected suggests that the use of chamber-specific myocytes will improve the accuracy and predictive nature of toxicity screens. And finally, chamber-specific myocytes also offer the opportunity for development of atrial fibrillation-specific medications or ventricular fibrillation-specific medications. And I just want to highlight that this work is done as part of a team that I'm very fortunate to lead at the University of Michigan with collaboration from scientists also at StemBioSys. And I want to thank you for your attention. And if there's any questions, I think Travis is going to help field those in the chat window.
That's right. Thank you so much, Todd. I think due to the timing, I think I'm going to forego kind of extra summary slides and jump right into the questions. I've received a couple through the chat, but I also have a few of my own, and so I'll read out the questions, and then hopefully Todd can field those for us. And so the first question was, is the maturation that you see quote unquote permanent? So will cells revert back to a fetal phenotype if they're replated from Matrix Plus back onto Matrigel?
That's a good question. We haven't tested that. We've just started to lift up the cells after they're matured to do patch clamp analysis, to do single channel recordings, but we haven't advanced that part of the project yet. So the answer is we don't know for sure.
Okay. So a lot in your workflows and the flow charts that you had, you always had the 7 days of culture on Matrix Plus followed by treatment. Can you go into how you decided to make day 7 the assay day?
That's based mostly on the morphology of the cardiomyocytes that we track using time-lapse imaging. So we know that when the cells are first plated from cryopreservation, all the cells are circular, and we've done some kinetic analysis to determine when the majority of the cells become rod-shaped and are contracting as a monolayer, and we found that to be 7 days.
So I've been really impressed by both the data that you showed today and some of the other data that I've seen in the past from your lab, looking at positive ionotropes and showing that we're able to see the positive ionotropic response consistently from not just isoproterenol, but other drugs with that effect. Is it known what features fetal-like cardiomyocytes lack that prevent them from exhibiting that response in vitro?
Yeah, they lack a mature sarcoplasmic reticulum. So the intracellular storage of calcium is immature. And also the link between the sarcolemmal membrane and the SR isn't highly organized as it should be in mature cells. And that can lead to dysfunction of the response to isoproterenol because the signaling cascade, the architecture likely just isn't there. Another reason for that effect could also be the use of metabolic stress media to mature cardiomyocytes. So the most popular method for purification of cardiomyocytes involves removal of glucose from media and substitution of glucose with lactate. In that system, only the cardiomyocytes, well, any cell with a large number of mitochondria will survive that kind of purification approach. And we've also shown that the cardiomyocytes respond to the ischemic media or to the lack of glucose and the substitution with lactate. They take on more of a heart failure-like molecular phenotype. And in heart failure, it's well known that there's dysfunction of the response to isoproterenol and other beta-adrenergic agonists.
Thank you. I think that's actually a perfect time for me to mention that this summer StemBioSys will be launching chamber-specific atrial cardiomyocytes that are iPSC derived as a for-sale product to complement our Matrix Plus product. If anyone that's on the presentation today is interested in being kind of a beta user for those, you can reach out directly to me at travis.block@stembiosys.com or our director of sales at iris.flores@stembiosys.com, or you can just visit our website and through the chat function, you can reach out and let us know that you're interested in being a beta user for our atrial cardiomyocytes. So I have a couple more questions, but again, if any of the attendees would like to ask a question, you can feel free to type it into the chat and we'll get it answered right now. Todd, you showed data for both acute and chronic toxicities. Are the common mechanisms for acute versus chronic toxicity going to be different? And so then, that leads me to, would we expect different types of drugs to result in false positive or false negatives in with fetal-like cells?
Yeah, so the difference between like mechanisms of toxicity being acute or chronic. So typically acute toxicity is detected using ion channel blockers because they act very rapidly and have very rapid effects on the function. On the other hand, chronic effects of medications, a good example of those is many chemotherapy drugs. They can be applied, and in the acute setting, like 1 hour to 2 hours, there's not really any observed cardiotoxicity or reduction in function, but days later, weeks later, there can be toxicity that becomes apparent. And that's likely due to the mechanism of action being mediated through the mitochondria. And the toxicity mediated through mitochondrial toxicity takes more time to become evident.
Interesting. That really shows the value of the mitochondrial data that you showed earlier as well, as far
Right,
as the
yes.
mitochondrial maturation.
Right. So any the importance of maturation there, as you said, is probably due to differences in the mitochondrial content, which we found to be much different between fetal cells and more mature adult cells. And so any mitochondrial toxicity, it's important that you have mature cardiomyocytes so that you can actually detect the toxicity.
So going deeper into that, one thing that has continued to surprise me about this work is actually how well the in vitro data has corresponded to known clinical risk. And the reason it surprised me is that I would have thought that there'd be more noise in the data due to biologic variability. Are there types of drugs that we would expect to need to test on more genetic backgrounds versus other types of drugs based on the mechanism of action?
Yeah, so again, another good example is chemotherapies. So it's known clinically that some patients can tolerate a relatively high dose of chemotherapy, and their heart function does not decline or become affected, while another patient receiving the same dose of a chemotherapy like doxorubicin can have very severe cardiotoxicity. So there can be patient-specific responses to drugs. And so for many drugs, it's important to increase the number of iPS cardiomyocyte lines that are used, just to be certain that what we're seeing isn't just an effect specific to that patient. So to have a broad number of iPSC lines and cardiomyocytes for testing is really the optimal way to do this kind of analysis and provide a more population-based kind of cohort.
Excellent. Thank you for that. So just one last question. So all the data today was about maturing healthy cardiomyocytes and getting kind of a healthy adult phenotype for toxicity testing. Can you just briefly comment on the potential of similar approaches for studying disease models?
Yeah, so one good example for the cardiomyocytes is the disease called hypertrophic cardiomyopathy, or HCM. This is a disease that's characterized by disorganization of the cardiac sarcomeres and also cellular disarray. And we've actually tested HCM patient-specific cardiomyocytes plated on Matrix Plus, and we could still observe that for the HCM patients caused by a genetic mutation, we could still see, even though the cells became rod-shaped, we could still detect the sarcomere disarray and also the cellular disarray relative to control cells. So yeah, the mature cells, still you can observe functional phenotypes as well as structural phenotypes that are expected in specific genetically linked cardiomyopathies.
Excellent. With that, I think that we have no more questions. So I just want to give a big thank you to my friend and collaborator, Dr. Todd Heron. It's always a pleasure to hear from you, and exciting to see your most recent work on the cardiomyocytes. For everyone that's still watching, this was a combination of published and unpublished data. But what's unpublished, you can expect to see published soon. Thank you all for joining today. And with that, I'll go ahead and end the presentation. Thanks again, Todd.
All right, thank you. Bye.